In the summer of 2005, food safety inspectors working across the interior highlands of Sardinia began compiling reports on a dairy product that had no legal status anywhere in the European Union. It was not a contaminated batch. It was not a spoiled shipment. It was a traditional sheep's milk cheese that had been made in the same mountain villages for a documented span of centuries, and the defining stage of its production involved deliberately exposing the wheel to a specific species of fly and allowing that fly to lay its eggs inside the paste. The larvae hatched. The larvae fed. The cheese was eaten with the larvae still alive inside it, and had been for as long as anyone in those villages could remember. The insect involved is Pyophila casei, the cheese skipper, a small dark fly whose larvae specialize in decomposing fat and protein, and which can propel themselves several inches into the air by seizing their own tail end and releasing under tension. A single mature wheel can contain thousands of them. The larvae do not simply live in the cheese. They are the production method. As they feed, they excrete enzymes that break the sheep's milk fats into a soft, weeping, almost liquid interior with an ammoniacal sharpness that no bacterial culture reproduces. The process takes 2 to 3 months. The wheel is scored, seeded, wrapped, and left in darkness, and what is removed at the end is not a cheese that has been infested. It is a cheese that has been digested. Roman agricultural writers described larval activity in aged cheeses without alarm, treating it as a stage of maturation rather than a failure of it. And the practice appears in variant forms across the Mediterranean, in Corsica, in parts of the Italian mainland, in Spanish and southern French mountain traditions, wherever sheep were kept on ground too poor for cattle, and a way had to be found to hold summer milk through winter. But here's where the conventional food safety framework stops working. The danger of this cheese is not the decomposition. Pyophila casei larvae are resistant to stomach acid. They are capable of surviving passage into the human intestinal tract, where they can attach to the intestinal wall and produce a condition called intestinal myiasis, presenting as abdominal cramping, bleeding, vomiting, and in documented cases requiring surgical intervention. This is not a theoretical risk, assembled by regulators looking for a reason to prohibit something. It is a recognized clinical entity with a case literature. The traditional response to it was procedural rather than pharmaceutical. Eaters were instructed to shield the cheese from the light so the larvae would not launch themselves toward the eyes. They were instructed to chew thoroughly and completely, because a larva that survives the mouth may survive everything after it. They were instructed to stop eating when the larva stopped moving, because dead larvae meant the cheese had passed into genuine putrefaction and was now carrying a different and less negotiable set of pathogens. The rule was simple and absolute. If it is not moving, it is not food. What makes this particular tradition unsettling is not the larvae, though the larvae are difficult to look at. It is what the rules surrounding them reveal about how the knowledge was held. Nobody in a 16th century Sardinian village understood microbiology. Nobody had a name for the enzyme cascade that turned hard curd into liquid. What they had instead was an inherited procedure with no explanation attached, a set of instructions passed from one generation to the next in which every step was load-bearing and none of the steps came with a reason. Follow the procedure and the result was a calorie-dense preserved fat source that could carry a household through a season of no grazing. Deviate from it, and the result was a substrate for one of the more efficient decomposition ecologies on the planet, sitting on the table, in the kitchen, beside the children. The margin between those two outcomes was measured in weeks and read by eye. Under European Regulation 853 of 2004, which governs the hygiene of food of animal origin, the cheese cannot be legally sold. Production continues anyway, informally, in kitchens and outbuildings and the back rooms of farms that do not advertise. The flies still find the wheels, the larvae still hatch in the dark, and the knowledge of exactly when to stop eating is now carried by a shrinking number of people who were taught it by someone who never explained why. Number 2. Hakarl. In 1968, a team of comparative physiologists analyzing the muscle tissue of Somniosus microcephalus, the Greenland shark, published a chemical profile that explained an 800-year-old North Atlantic culinary practice and simultaneously made it look far more alarming than it had ever looked before. The Greenland shark is a slow, enormous, deep-water predator that can exceed 20 feet in length and which recent radiocarbon analysis of eye-lens proteins suggests may live for more than 300 years, making it the longest-lived vertebrate ever identified. It is also, in the ordinary sense of the word, inedible. Its flesh is saturated with urea and trimethylamine oxide at concentrations that allow the animal to maintain osmotic balance in cold saline water at depth without the energy cost of active regulation. Consumed fresh, that chemistry does not stay inert. Gut bacteria and endogenous enzymes convert trimethylamine oxide into trimethylamine and urea into ammonia, and the result in a human being is a syndrome that Norse-descended fishing communities described for centuries in terms indistinguishable from acute intoxication. Disorientation, loss of motor coordination, vomiting, violent gastrointestinal distress, insufficient quantity, convulsions, and death. Sled dogs fed fresh Greenland shark have been observed to lose the ability to stand. And yet the animal was hunted deliberately, in numbers, across Iceland, Greenland, and coastal Norway for the better part of a millennium, because in a subarctic economy with almost no arable land, a single shark represented several hundred kilograms of protein and oil that could be secured with a hand line from a small boat in winter. The processing solved a problem that fire could not. Heat does not remove urea. Boiling does not remove trimethylamine oxide. What removes them is time, pressure, and a controlled microbial succession that the practitioners could not describe but could reproduce with precision. The shark was gutted, beheaded, and placed in a shallow pit dug into gravel above the tide line, then covered with sand and weighted with stones. The pressure drove fluid out of the tissue over a period of 6 to 12 weeks. The mass was then exhumed, cut into strips, and hung in an open ventilated shed exposed to sea air for a further 4 to 5 months. Over that combined period, bacterial and enzymatic action reduced the urea load to a survivable concentration and drove off much of the volatile amine content as gas. What was left was a pale, fibrous, ammonia-heavy protein block that could be stored without refrigeration for a year. But here's where the standard account of this as a quaint regional oddity falls apart. There is no visible endpoint to the process. There is no color change that signals safety, no texture that reliably indicates the urea has fallen below threshold, no smell that distinguishes adequately cured tissue from tissue that will still put an adult on the floor. The measurement instrument was the calendar and the season, and both of those were themselves proxies for temperature and humidity conditions that varied year to year. A mild winter shortened the effective fermentation. A wet spring extended it. The practitioners compensated by holding the schedule conservatively and by testing in small quantities on the household's own members before the batch entered general consumption. The safety margin was built out of caution, repetition, and a willingness to be the first person to find out. What makes this practice genuinely unsettling is not the toxicity. It is the length of the feedback loop. A cook who undersalts a stew learns within minutes. A community that mistimed a shark pit learned 5 months later, at the moment of consumption, with no ability to correct the batch and no way to know whether the error was in the burial depth, the gravel composition, the ambient temperature, or the size of the animal. Every correction to the method had to be inferred backward across half a year from an outcome that might have had a dozen causes. Refining that procedure to reliability would have required generations of accumulated failure, and every data point in that dataset was a person who got sick. The pits are still dug. The sheds still stand along the same stretches of coast. The processing time has not been shortened by a single week in 800 years, because nobody has ever found a way to make the animal safe faster, and the people who worked out the original schedule left no record of how many attempts it took. Number 3. Bitter almond. In 1922, when the antechamber of a New Kingdom Egyptian royal burial was catalogued, the inventory of provisions interred with the body included baskets of almonds, placed alongside dates, chickpeas, and grain as food intended to sustain the occupant through a journey the culture regarded as literal. Almonds appear in Egyptian, Levantine, Mesopotamian, and Greek food records continuously from the Bronze Age onward. They were traded, taxed, pressed for oil, ground into pastes, and buried with the dead. They were also, in their original wild form, one of the most efficiently lethal plant foods in the human diet. Wild almond, Prunus dulcis in its bitter expression, contains amygdalin, a cyanogenic glycoside stored in the kernel as a chemical defense. Amygdalin is stable and harmless on its own. What makes it dangerous is that the same seed contains, in separate cellular compartments, the enzyme that breaks it down. Crush the kernel, or chew it, and the compartments rupture. The enzyme meets the substrate. The reaction products include benzaldehyde, which produces the distinctive marzipan odor, and hydrogen cyanide, which binds cytochrome C oxidase and shuts down the ability of the body's cells to use oxygen. Death occurs in a person whose blood is fully oxygenated and who is nonetheless asphyxiating at the cellular level. The numbers are brutally small. A bitter almond kernel can carry between 4 and 9 milligrams of releasable hydrogen cyanide. An adult lethal dose sits in the range of 50 to 60 milligrams. Between 10 and 20 kernels can kill a grown man. For a child, the figure has been estimated at as few as 5. The same chemistry appears across the entire Prunus genus, in apricot kernels, peach pits, cherry stones, and plum pits, all of which were eaten deliberately in the ancient world, ground into flavoring pastes, pressed for oil, steeped into drinks, and used medicinally by physicians who described their effects with unmistakable clinical accuracy centuries before anyone isolated the compound responsible. But here's where the story of ancient food safety becomes something closer to a story about genetics. Sweet almonds exist because of a mutation. A single dominant allele suppresses amygdalin production in the kernel, and trees carrying it produce seeds that can be eaten in quantity without consequence. That mutation is rare in wild populations, and it is not stable across sexual reproduction, because an almond tree grown from a sweet almond seed frequently reverts. The only way to hold the trait is vegetative propagation, grafting cuttings from a known safe individual onto rootstock, which means every sweet almond eaten in the modern world descends from a small number of original mutant trees identified by someone in the Levant or Central Asia at least 5,000 years ago. Somebody found a tree whose seeds did not kill people. Somebody understood that the property could not be transmitted through the seed and had to be transmitted through the wood. Somebody worked out how to graft. And somebody, before all of that, established which trees were safe, and the only available method for establishing that was to eat from them. What makes this particularly disturbing is not the poison, it is the sorting problem. A bitter almond and a sweet almond are visually indistinguishable. Same shell, same shape, same size, same color. The only reliable field test is taste, and the bitterness that signals danger is detectable in a quantity well below the lethal dose, which means the safety mechanism available to ancient harvesters was to put the thing in the mouth and find out. Across the growing regions of the ancient Mediterranean and Near East, orchards were a mosaic of grafted safe stock and volunteer seedlings that had reverted, and the difference between a snack and a fatal dose was carried entirely in the sensory judgment of whoever was picking. The practice of eating bitter kernels never stopped. They remain in use as flavoring across multiple cuisines, in trace quantities, and they remain in circulation as a supposed therapeutic under a marketing name for a compound with no demonstrated benefit and a well-documented poisoning literature that grows every year. The orchards are still there. The grafted trees still produce. And somewhere in every wild almond population, the ancestral allele is still expressed, still stored in 2 compartments, still waiting for something to break the cell wall. Number 4. Tarwe. In 1963, agronomists surveying highland cultivation across the Peruvian and Bolivian Altiplano documented a legume being grown at elevations above 11,000 feet, where almost no other protein crop would set seed. Lupinus mutabilis, known locally as tarwe, produced beans containing between 40 and 50% protein by dry weight, a figure that exceeds soybean, alongside a substantial oil fraction. In a landscape of thin air, brutal night frost, and marginal soil, it was one of the most nutritionally efficient plants ever brought under domestication in the Americas. Archaeological recovery has placed lupin seeds in Andean burial contexts and coastal middens dating back more than 2,000 years, and depictions of the plant appear in the ceramic and textile record of multiple pre-Columbian cultures. It was a staple. It was also, in its harvested state, completely inedible. The problem is quinolizidine alkaloids. Bitter lupin varieties concentrate a family of compounds including lupinine, spartine, and related molecules that act on the nervous system as sodium channel blockers and anticholinergics. Ingestion of unprocessed seed produces dry mouth, blurred vision, dilated pupils, disorientation, motor impairment, elevated heart rate, and at higher doses, respiratory depression and cardiac disturbance. Alkaloid content in bitter varieties can reach 3 to 4% of seed dry weight. A modest handful of raw beans is enough to hospitalize an adult. Spartine in particular has a documented cardiac effect substantial enough that it was used pharmaceutically in the 20th century as an antiarrhythmic and as a labor-inducing agent before safer compounds displaced it. The Andean solution was a processing chain that took the better part of a week and consumed a resource that the highlands could not always spare. The beans were boiled, then transferred into porous woven sacks, and the sacks were submerged in a stream, weighted, and left in the current for 4 to 7 days. Because the alkaloids are water-soluble and the protein is not, the running water pulled the toxic fraction out of the seed while leaving the nutritional value substantially intact. The end point was determined by taste. When the bitterness was gone, the beans were done. A parallel tradition developed independently around the Mediterranean, where Lupinus albus was processed by repeated soaking in salted or seawater and became a common food across the Roman world, sold cheaply enough that it carried a persistent association with poverty and appeared in comic drama as the food of the destitute. But here's where a simple leaching procedure turns into something with a much narrower margin than it appears. The taste test is not a reliable proxy for safety at the boundary. Alkaloid perception varies between individuals, and a batch that reads as acceptably mild to one person can carry a clinically significant load for a smaller person, a child, or someone processing it slowly. 20th century poisoning clusters in Europe, in South America, and in the Middle East have overwhelmingly involved lupins that were partially debittered and eaten by people who assumed that partial processing was proportionally safe. It is not. Symptoms appear abruptly once a threshold is crossed, rather than scaling smoothly with dose, which means the sensory warning and the physiological consequence are not calibrated to each other. What makes this genuinely unsettling is not the chemistry. It is the infrastructure requirement buried inside the recipe. This food cannot be made without a reliable flow of clean, cold, moving water for a continuous week. It cannot be made in a drought. It cannot be made during a siege. It cannot be made by a family that has been displaced from its watercourse, or by a community whose stream has been diverted, or by refugees carrying seed but not carrying access to the specific hydrological conditions their entire preparation method assumes. The nutritional value of the crop was never really contained in the crop. It was contained in the relationship between the crop and a piece of running landscape, and anyone who kept the seed but lost the stream kept a sack of neurotoxin. The plants still grow above 11,000 feet, the streams still run. Sweet cultivars bred in the 20th century now exist with alkaloid levels below 0.1%, and they cross freely with bitter stalk in any field where both are present, producing seed that looks identical to both parents and tastes like nothing at all until the moment it doesn't. Number 5. Nardoo. In 1994, 2 Australian researchers publishing in a general science journal presented a chemical analysis that closed a case which had been open for 133 years. In 1861, an expedition attempting to cross the Australian continent from south to north had disintegrated on its return leg. 3 men reached a camp on a creek in the central interior, found it abandoned, and survived for a period of months on food, obtained locally, with the assistance of the Aboriginal people of the region. 2 of the 3 died of what contemporaries recorded as starvation, despite eating substantial quantities of a locally abundant food, and despite one of them writing, days before his death, that he was consuming enough to feel full, and was nonetheless growing weaker by the hour. The third man survived, having remained in the company of the local community and eaten what they ate, prepared the way they prepared it. The food was nardoo, the sporocarp of Marsilea drummondii, an aquatic fern that carpets the floodplains of arid inland Australia and produces hard, pea-sized reproductive bodies in enormous quantity when the water recedes. It had been a staple across the interior for thousands of years. Grinding stones with nardoo residue have been recovered across a vast geographic range, and the plant provided a dependable carbohydrate source in a landscape where dependable anything is rare. The processing was specific. The sporocarps were roasted, then ground on a stone into a coarse flour, then mixed with water and either baked into a cake or cooked into a paste, and critically, the flour was worked with water carried in a vessel and heated rather than left raw. The chemical analysis identified the reason the sequence mattered. Nardoo contains thiaminase, an enzyme that cleaves thiamine, vitamin B1, and destroys it. Consumed raw or inadequately heated, nardoo does not merely fail to provide nutrition. It actively strips the body of a vitamin it needs to metabolize carbohydrate at all. The result is beriberi, presenting as peripheral neuropathy, muscle wasting, loss of reflexes, confusion, cardiac involvement, and a progressive collapse that mimics starvation exactly while the sufferer continues to eat. The more nardoo consumed, the faster the depletion. The men who died were killed by the volume of food they were eating. But here's where the story stops being about an expedition and becomes something more uncomfortable. The Aboriginal communities of the region had been eating nardoo without incident for a period measured in millennia, and the difference was not luck. It was procedure. The roasting step degrades thiaminase. The preparation of the flour with water and heat degrades more of it. The consumption of nardoo, as one element within a broader diet that included fish, mussels, seeds, and small game, supplied thiamine from other sources that offset whatever enzyme survived. The expedition members observed the food, but not the method. They saw people eating a ground flour cake, and they replicated the visible half of the operation. They ground the sporocarps. They mixed the flour with cold water. They ate it in quantity as their sole subsistence, and they wrote in their journals that they could not understand why the food was not sustaining them. What makes this particularly disturbing is not that they got it wrong, it is that a food preparation method can be almost entirely invisible from the outside. The dangerous steps in this process are not dramatic. There is no ceremony, no obvious ritual, no gesture that an observer would flag as load-bearing. A roasting stage that looks like ordinary cooking, a water temperature that looks like ordinary mixing, a dietary context that looks like ordinary variety. Every element that separated a staple from a slow poison would have been invisible to someone watching from 20 feet away and taking notes, which is exactly what happened. The floodplains still flood, the fern still carpets them when the water pulls back, and the sporocarps still come up in the millions, and the thiaminase is still in every one of them. The grinding stones are still lying where they were left, on the same creek beds, holding residue that has been dated across thousands of years of correct preparation by people who never once needed to write the instructions down. There are 5 more of these, and the last one killed more people than everything else on this list combined. Subscribe now and stay for it. Number 6. Cycad flour. In the mid-1950s, epidemiologists working for the United States National Institutes of Health began investigating a cluster of neurodegenerative disease on Guam that was without precedent anywhere in the world. Among the indigenous Chamorro population, rates of a syndrome combining features of amyotrophic lateral sclerosis, Parkinsonism, and dementia were running at somewhere between 50 and 100 times the global background rate. In some villages, it was the leading cause of adult death. Families lost multiple members across a generation. The condition, designated ALS PDC, had no infectious agent, no clear inheritance pattern, and no environmental exposure that immediately explained it. The investigation would run for more than 6 decades and remains unresolved, but the leading hypothesis has centered from early on around a food. The food is cycad flour. Cycads are among the oldest surviving seed plants on Earth, a lineage older than flowering plants, and their large starchy seeds have been used as a famine and staple carbohydrate across the tropical Pacific, Southeast Asia, Australia, and Southern Africa. On Guam, the seeds of Cycas micronesica were husked, sliced, and washed repeatedly in changes of water over a period of days to weeks, then dried and ground into a flour used for tortillas, dumplings, and thickening. The washing was not optional, and everyone knew it. Cycad seed contains cycacin, a potent hepatotoxin and carcinogen, and the acute effects of inadequately processed cycad are immediate and unmistakable, producing violent vomiting, liver damage, and in livestock a distinctive hind limb paralysis that gave the syndrome its stockman's name. The traditional leaching protocol was refined enough that acute cycad poisoning was rare among people who followed it. The processing worked, for the thing it was designed to remove. But here's where the standard model of food safety, which assumes that harm announces itself, comes apart entirely. Cycad tissue also contains a non-protein amino acid, designated BAMA, produced not by the plant itself, but by cyanobacteria living symbiotically in specialized coralloid roots that grow upward out of the soil. BAMA is water-soluble in part, but researchers working from the 1990s onward, most prominently an ethnobotanist working with Pacific Island communities and a neurologist who documented the Guam cases in detail, proposed that it bioaccumulates. It concentrates up the food chain. It was present not only in the washed flour but in the flying foxes that fed heavily on cycad seed and which were themselves a prized ceremonial food, consumed whole. And BAMA does not act in hours or days. It has been proposed to misincorporate into proteins in place of serine, producing misfolding that accumulates in neural tissue across decades before any symptom appears. If the hypothesis is correct, the exposure that killed a person at 60 occurred when they were 20. What makes this genuinely terrifying is the collapse of the feedback mechanism that every other traditional food safety system on Earth depends on. Every preparation method humans have ever developed for a dangerous food was built by correlating an input with an outcome. Eat this, get sick, change the method. Do not get sick, keep the method. Teach it to the children. That entire architecture requires the outcome to arrive close enough to the input that a connection can be drawn. Stretch the interval to 30 or 40 years, and the mechanism does not merely weaken, it stops functioning. No amount of accumulated cultural wisdom, no number of generations of careful observation, and no degree of intelligence on the part of the practitioners can identify a cause that is separated from its effect by half a lifetime. The community was not careless. The community was operating with a detection system that cannot see this class of harm at all, and neither, for most of its history, could ours. Incidents on Guam declined sharply across the second half of the 20th century as dietary patterns shifted, which is consistent with the hypothesis without proving it, and the debate over BMI continues in the literature with genuine disagreement about mechanism and dose. Cycads still grow across the Pacific. The seeds are still processed and eaten in multiple countries. The leaching protocols still remove what they were designed to remove, and nobody in the 19th century had any way of knowing that a food could be prepared correctly, eaten safely, and still be counting. Number 7. Bitter cassava. In the early 1980s, a Swedish physician working in a rural district of northern Mozambique during a period of drought and armed conflict began documenting an outbreak of sudden, permanent, symmetrical paralysis of the legs. It appeared abruptly, often within hours, overwhelmingly in children and women of childbearing age, and it did not progress and did not improve. Hundreds of cases accumulated. The local name for the condition described the tied, spastic gait it produced. There was no pathogen, no vector, and no evident deficiency that the standard investigative playbook could identify. What the affected households had in common was that they had been eating bitter cassava that had been processed in a hurry. Cassava, Manihot esculenta, feeds an estimated 800 million people. It is the most productive calorie crop per unit of marginal land ever domesticated. It tolerates drought, poor soil, and neglect. It can be left in the ground as a living granary for 2 years after maturity, and it was brought into cultivation in the southwestern Amazon at least 8 to 10,000 years ago. It is also, in its bitter varieties, loaded with cyanogenic glycosides, principally linamarin, at concentrations that make the raw root reliably lethal. As with the almond, the toxin and the enzyme that activates it are held apart within the intact tissue, and as with the almond, damage to the tissue brings them together and liberates hydrogen cyanide. The bitter varieties are not an accident or a primitive stage awaiting improvement. They are preferred in many growing regions precisely because the toxicity deters insects, rodents, wild pigs, and thieves, which means the farmers selected for the poison deliberately and then built the detoxification into the kitchen. The processing chain developed in the Amazon and carried across the Atlantic is among the most elaborate food preparation sequences ever devised. The roots are peeled, then grated or pounded, to rupture the cells and force the enzyme into contact with the glycoside, which begins the release of cyanide before the food is anywhere near a person. The mash is then packed into a woven press, in some traditions a long plated tube that elongates under load, and squeezed for hours to expel the liquid fraction carrying the dissolved toxin. The pressed solids are left to ferment, then sieved, then toasted over a fire with constant agitation, driving off the remaining volatile cyanide as gas. The process can span 3 to 6 days from harvest to edible flour. Every step removes a portion. No single step removes enough. But here's where the elegance of that solution becomes something much darker. The chain has no redundancy under stress. Each stage assumes the previous stage completed, and the compounding effect that makes the sequence safe also means that shortening it does not produce a proportionally smaller risk. It produces a residue that is still well within the range that causes harm. Under famine, under displacement, under the specific conditions of a war that separates people from their equipment and their time, the pressing gets cut short, the fermentation gets skipped, the toasting gets abbreviated because fuel is scarce, and the resulting flour looks and cooks and tastes close enough to correct. Chronic subacute cyanide exposure in a population that is simultaneously protein deficient and therefore short of the sulfur amino acids the body uses to detoxify cyanide produces exactly the irreversible motor neuron damage that was documented in Mozambique and subsequently across a belt of Central Africa. The disease is a disease of interrupted procedure. What makes this the most quietly disturbing entry on this list is the direction of the causation. The toxin does not strike when the crop fails. The toxin strikes when the crop succeeds and everything else fails. Cassava is the food people fall back on when the rest of the system has collapsed, which means the moment of maximum reliance on it coincides precisely with the moment of minimum capacity to process it correctly, and the plant that keeps a population alive through catastrophe cripples a fraction of them on the way through. Simplified processing methods now exist, developed in the last few decades, that reduce residual cyanogens using nothing but wetting and shade. They are cheap, they are effective, and they are still not universally known in the regions that need them most. The fields are still planted, the presses still hang, and the roots still sit in the ground, patient and productive and holding, in every cell, both halves of a reaction waiting to be completed. Number 8. Grass pea. In 1863, a Spanish printmaker completed a series documenting a period of war, occupation, and starvation, and one of the plates in that series showed a group of figures queuing for a distribution of flour. The caption thanked the grain for the fact that they were eating it, and the tone of the thanks was not straightforward, because everyone looking at the print in Spain at the time knew what the flour was and knew what it did. The grain was almorta, the grass pea, Lathyrus sativus, and by the middle of the 20th century, following another Spanish famine, the same crop would produce thousands of permanently disabled survivors and would be banned for human consumption by a government that had spent the preceding years distributing it. Grass pea is the most stubbornly useful legume ever domesticated. It has been cultivated across the Mediterranean, the Middle East, the Horn of Africa, and South Asia for at least 8,000 years, with charred remains recovered from Neolithic settlement layers across a wide geographic span. It fixes its own nitrogen. It carries protein content in the range of 25 to 30%, and it has a drought tolerance that borders on the absurd, producing a harvest in conditions where wheat, barley, chickpea, and lentil have all failed completely, while simultaneously surviving waterlogging that would rot most other pulses in the ground. It is, functionally, an insurance policy planted at the field margin, ignored in good years, and harvested in the years when there is nothing else. It has never needed marketing. It has only ever needed a disaster. The cost is a compound called beta-ODAP, a non-protein amino acid that acts as an excitotoxin at glutamate receptors in the central nervous system. Consumed as a minor component of a varied diet, it produces no detectable effect. Consumed as the dominant or sole source of calories for a period of roughly 2 to 4 months, it produces neurolathyrism, an irreversible spastic paralysis of the lower limbs caused by degeneration of the upper motor neurons that control them. It arrives without warning. There is no prodrome that reliably signals the approaching threshold. A person walks normally, and then over a period of days develops stiffness, then a scissoring gait, then dependence on a stick, then on 2 sticks, then on the ground. The damage does not reverse. Young men doing heavy physical labor on a grass pea diet are the most affected group, which in a famine is precisely the group a household cannot afford to lose. But here's where the situation becomes something other than a simple story of a toxic plant. Traditional preparation methods that reduce ODAP have existed for as long as the crop has. Prolonged soaking with discard of the water, boiling in excess water that is then thrown away, fermentation, and roasting all lower the concentration substantially. The knowledge is not lost. It is not secret. It is not even complicated. What happens in a famine is that every one of those methods costs something the household no longer has. Soaking costs water. Boiling in excess water and discarding it costs both water and fuel, and throws away dissolved calories that a starving family cannot spare. Fermentation costs days. The correct preparation of the emergency food requires resources that are only ever absent under exactly the conditions that make the emergency food necessary, and so the crop is eaten in the way that maximizes short-term survival and guarantees a proportion of long-term disability. What makes this the most bitterly designed food on this list is that the trade is not hidden. Communities that grow grass pea have generally known what it does. Physicians described the syndrome in antiquity, and the association between the crop and the paralysis appears in medical and agricultural texts across 2,000 years and multiple languages. Nobody was fooled. Farmers planted it anyway, because the calculation is genuinely unresolvable. A crop that might cripple some fraction of the people who rely on it is superior, in a failed year, to no crop at all, and every household that has ever planted it has made that arithmetic explicitly and accepted the terms. It is still planted across millions of hectares. Low ODAP varieties have been bred and released and have struggled with adoption, partly because the trait tends to come paired with reduced hardiness, which removes the only reason to grow the plant in the first place. The fields still go in at the margins. The insurance policy still sits there through the good years, unharvested and unremarked. And in the bad ones, it does what it has always done, and takes what it has always taken. Number 9. Defrutum. In 1983, an environmental geochemist published an analysis of ancient dietary practice, arguing that the ruling class of a Mediterranean empire had been subject to chronic exposure to a heavy metal at levels sufficient to produce measurable cognitive, reproductive, and behavioral effects across generations. The claim was contentious and remains partially contested, but the underlying chemistry was never in dispute, and subsequent sediment work, published 3 decades later, analyzing lead isotope signatures in the harbor deposits of a major imperial river, confirmed a lead contamination signal in the region's water supply that tracked the rise and fall of imperial infrastructure with uncomfortable precision. The vector at the center of the original argument was not the plumbing. It was a food. The food was defrutum, and its more concentrated relative sapa, a grape must syrup produced by boiling unfermented juice down to a fraction of its original volume. It functioned as the empire's principal sweetener in a world with no accessible cane sugar, and it appeared everywhere. It sweetened wine. It preserved fruit. It was added to sauces, to stews, to medicines, and to cosmetics. Agricultural manuals of the period specify the preparation in detail, including the crucial instruction that the reduction should be carried out in a vessel of lead, or of lead-lined bronze, rather than in copper. The instruction was not arbitrary, and it was not ignorant. Copper vessels imparted a metallic bitterness and a green discoloration to acidic grape must. Lead vessels did not. Lead vessels made the syrup taste better. The reason they made it taste better is that the acid in the must reacted with the metal to form lead acetate, a compound with a pronounced sweetness of its own, which meant the vessel was not merely a neutral container, but an active ingredient. Modern reconstructions boiling grape must in lead-lined vessels using the documented method have produced syrups with lead concentrations in the range of hundreds to thousands of milligrams per liter. The recommended daily consumption implied by surviving recipes, spread across wine and preserved fruit and seasoning, would have delivered a dose that exceeds every modern occupational exposure limit by orders of magnitude. Lead accumulates. It deposits in bone with a biological half-life measured in decades. It crosses the placenta. Its effects at chronic low dose include anemia, gout, infertility, miscarriage, elevated aggression, impaired impulse control, and reduced cognitive function. And it does all of this without producing a single symptom that would ever be attributed to the sweetener. But here's where this stops being a story about an ancient mistake and becomes something considerably more unsettling. The exposure was not distributed randomly. It was distributed by wealth. The people consuming the most sweetened wine, the most preserved fruit, the most elaborately seasoned food, and the most cosmetic preparations were the people at the top of the social structure, which means the population most heavily dosed with a neurotoxin known to degrade judgment, fertility, and emotional regulation was the population making decisions about law, war, succession, and the allocation of an empire's resources. Skeletal lead analysis from imperial period burials has repeatedly shown elevated body burdens, with the highest concentrations in remains from higher status contexts. Whether that explains anything about the historical record is a question historians have argued over for 40 years and will keep arguing over, because the causal chain is unprovable and the temptation to reach for it is obvious. What is not arguable is the exposure itself. What makes this genuinely disturbing is not that they poisoned themselves. It is that the poison arrived as an improvement. Nobody was forced to use lead vessels. There was no shortage of alternatives, no technological limit, no ignorance of metallurgy. The method spread because it produced a better product, because the syrup was clearer and sweeter and kept longer, and because every individual cook comparing the two outputs side by side would have reached the same conclusion and made the same choice. The optimization was real, the feedback was immediate and positive, and the cost was invisible, delayed, cumulative, and indistinguishable from ordinary human frailty in a society with a high baseline of illness and death. The recipes survive. The vessels have been excavated. The isotope signal sits in the harbor sediment where it has sat for 2,000 years, rising with the aqueducts and the mints and the mines, falling when the infrastructure did, a chemical record of an empire's appetite written into mud. Number 10. Nixtamal. In 1735, a physician serving a royal court in northern Spain described a disease he had been documenting among the rural poor of his province, characterized by a symmetrical scaling rash on sun-exposed skin, chronic diarrhea, progressive dementia, and death. He called it by a local name referring to the roughness of the skin. Over the following 2 centuries, the same disease appeared across northern Italy, southern France, the Balkans, Egypt, southern Africa, and eventually the southern United States, where by the 1920s it was producing tens of thousands of cases annually and killing thousands of people a year in institutions, mill towns, tenant farms, and orphanages. The condition was pellagra, and every location where it appeared shared a single characteristic. All of them had recently adopted maize as a dietary staple. None of them had adopted the processing method that came with it. Maize was domesticated in Mesoamerica from a wild grass and became the foundation of every major civilization in the region. Alongside the crop, those cultures developed a technique in which dried kernels are simmered and then steeped in an alkaline solution made from wood ash or slaked lime. The kernels swell, the outer pericarp loosens and is rubbed away, and the softened grain is ground into a dough. The The technique is documented archaeologically through residue in cooking vessels and the form of the grinding equipment, with evidence pushing it back at least 3,000 years. To an outside observer, the alkaline step looks like a mechanical convenience. It removes the hull, it softens the grain, and it makes the dough cohesive enough to shape by hand. Every visible function of the step could be explained without reference to nutrition at all. But here's where the entire history of the last 500 years of global agriculture turns on a chemical detail that nobody involved could have known. Maize contains niacin, vitamin B3, in an ample quantity, but it is bound in a form called niacin that the human digestive system cannot liberate. It passes through unabsorbed. Alkaline treatment hydrolyzes that bond and releases the niacin into a bioavailable form. It also increases the availability of the amino acid tryptophan, which the body can convert into niacin on its own, and it improves calcium content substantially by depositing it from the lime. A population eating alkali-processed maize gets everything it needs. A population eating the identical grain from the identical seed stock, prepared by simple grinding and boiling, is niacin deficient, and sustained niacin deficiency produces exactly the dermatitis, diarrhea, and dementia that were cataloged across 3 continents for 200 years. When maize crossed the Atlantic in the 16th century, it crossed as a seed and as an agronomic package. The yields were extraordinary. It grew where the established grains struggled, it produced more calories per acre than wheat or rye, and it was adopted fastest by exactly the populations with the least margin: tenant farmers, subsistence smallholders, the institutionalized, and the enslaved. What did not cross was the kitchen. The lime, the ash, the steeping, the specific vessels, and the specific timing were a domestic practice performed by women in households, transmitted by demonstration rather than by text, invisible in the account books and cargo manifests and agricultural treatises that the transfer actually ran through. The people who moved the crop moved the part of the system that was written down. What makes this the heaviest entry on this list is the accounting. It took until the 1930s for a public health investigator in the American South to demonstrate, against enormous institutional resistance, that pellagra was dietary rather than infectious. In the intervening centuries, the disease killed an unknown number of people, plausibly in the millions, across every region that adopted the grain without the method. Not one of those deaths was caused by a dangerous food. The grain was never the problem. The problem was that a procedure with no visible purpose, whose function could not be deduced from watching it, and could not be justified by anyone performing it, was classified as a local custom and left behind at the point of transfer. The fields are still planted, on a scale that dwarfs anything the crop's domesticators could have imagined. The alkaline method is still used across its region of origin, unchanged in its essentials, still producing the same dough from the same steps. And the instruction that would have saved those centuries was never lost, never hidden, and never secret. It was simply never asked for, because nobody taking the seed believed there was anything left to ask. If you want to see more videos like this, click the video on screen now, and make sure to subscribe.